So I'm really pleased to see so many people here for this special division seminar. This is both an internal seminar, but also, I guess, a public seminar for the rest of the Institute. Now, before I begin, of course, I'd like to begin by acknowledging the traditional owners of the lands on which we meet today, the Wurundjeri people of the Kulin Nation, and also to acknowledge any Aboriginal First Nations people who work in our Institute, who contribute so much to our science. I'm really excited to introduce Professor Hal Drakesmith. So to get the obvious out of the way first, Hal was my postdoc supervisor. I spent 5 years in Hal's lab between 2012 and 2017. And surprisingly, he still had a job after I left. I don't know, tried my best to damage the place, but anyway. Hal did his PhD in Cambridge, I think. Was that right?
I can't remember.
Yeah. And was also at UCL prior to that. But and so he really downgraded when he came to Oxford. But you know, these jokes don't work so well here. I think they don't really work. Yeah. Actually, just to think about how you sound when you say stuff like that, probably not so good. But anyway, yes, if we're in England, this is the first 20 minutes of any talk. But he set up and run a highly successful lab that studies iron and ... That could be the summary of it, but the principles and the priorities of what they work on is, of course, around immunology. He belongs to the MRC Human Immunology Unit. And some of the really big achievements that his lab have made over the last few years have included discovering how erythroferrone, which our lab partly works on as well, how it actually works, how it actually acts on the liver to cause that effect on its target. And that's a seminal contribution that is being cited by the second. He's made many other contributions around how viral infections influence hepsin regulation, and thereafter a whole new, or a pretty big new pathway around how intracellular sensing of inflammation and viral infection. I'm stuffing this up. How that works, and including in hepatitis C. But more recently, his lab has pivoted or expanded into the area of iron and immunology, and he's made some very important contributions there that were really just sort of starting to take off as I was leaving, probably because I left. The lab has become far more successful. So I'm really, and more than that, actually, I'll just add one other thing, which is that Hal, one of the things he really taught me was, see, I've talked so long that the screen has stopped working, but you know, that's fine. One of the other really important things about working with Hal, and I think he's a brilliant speaker, well, just to put the pressure on. But secondly, you know, his lab was just an amazing place with a great culture, and I think that was one of the most important things that I brought back when I got to set up my own lab was the value and importance of a lab that can work together and works well as a team. And so I think you'll hear some of not just the science, but some of the way that Hal's team approaches their science in this talk. Hal, all yours.
Thanks for the introduction, Santa. I don't know whether I was obviously not going to be nice to you, but I'm definitely now not going to be nice to you. I think that's going to be good. So this is fun. No, seriously, it's an honor to be here. It's really great to be here. Thank you so much for inviting me. I'm going to be talking about hepcidin and iron, and here we have T cells in the middle and then transferrin on the right. But really, primarily, I just want to start off just by saying thanks to Santa for the invitation, for looking after me, but also thanks for being in my lab. You know, you were unbelievably productive when you were in my lab. There were so many projects going on, and when it didn't matter what it was, whether it was molecular control of hepcidin or global health or new mechanisms of how erythroferrone might work, Santa was just always really excited and really interested, and you know, he was unstoppable. This is one of my favorite photos. And then, but obviously, he did do great stuff. And as a result of really Santa's analysis of how Hepsin was working in global health situations, we got a really nice grant to work out in the Gambia. And one of the greatest things about that is we could buy a Land Rover. So that's for Santa and Andy Armitage, who I'll talk a bit about as well, and the Land Rover that we got. But, lab culture is really important to make a lab work. And Santa was superb at contributing to the lab culture. So this is really a great picture. One of the things that's important to notice here is the intellectual brilliance of knowing you've got a really big moustache and you're drinking a pint of Guinness. How are you going to do that? You put a straw in the Guinness. Okay, that's working around a situation. Clearly the only way is up from there. But to put it into context, of course, Santa was basically joining in the situation of what was occurring. And I've even forgotten what on earth this was about, but there are all sorts of people doing all sorts of things here. So thanks very much, Santa. Santa, I've actually got you a couple of things. So one of the things that Santa was great at and really added to the lab that we weren't very great at was good statistical analysis. Now, you might laugh now, but it's true. He was really great at statistical analysis. So I've got him a book called Things that he was really good at dealing with. It's called The Outliers. So you've got that, and then Santa's kind of going up in the world of the, it's all going really well. So he's going to have to become a bit less scruffy, maybe. So I've got him a tie, and it's a tie with what looks like gametocytes on it as well. So Santa, do you want to? Can I give you these? Okay, so. Now I'll give my talk, and a bit of an immediate change of tone. So, plague in Chicago. This was a remarkable occurrence, very sad occurrence in many ways. It was an unexpected death of a guy called Malcolm Casadaban, who was a professor of microbiology who worked at Chicago University. And he was admitted with sepsis, but they couldn't do anything with him, and he died about half a day after being admitted. And so this had to be investigated, primarily because of what he was working on. And he was working on Yersinia pestis, the causative agent of the Black Death. But it's thought that the only version that he was working with was a highly attenuated version, which had great chunks of its genome deleted and had been used in category 2 safe conditions all over the planet for decades and decades. It had never caused any trouble at all. However, the wild type, deadly version, was in use in that institute that he was working. So the suspicion was there'd been some sort of contamination, some sort of leak, as can happen. And so everything got locked down, everybody was quarantined, everybody had to get tested and so on. But eventually, post-mortem blood cultures from Professor Casadaban revealed that, in fact, he was infected with the attenuated strain, which should have been completely harmless. So it was very mysterious how that could happen, until eventually the physicians were able to diagnose him, again, post-mortem from autopsy results, with hemochromatosis. So he had undiagnosed iron overload, and the key observation here was that the reason why that attenuated strain of Yersinia pestis was safe was because those great chunks of the genome that were deleted were encoding high affinity iron scavenging machinery. That means that the wild type is incredibly good at scavenging iron from the host, but the attenuated version lacking that isn't, which is why it's relatively safe. However, that defect in the bug was complemented by the genetic lesion in Professor Casadaban, showing that despite having an active immune system, because the iron level was wrong, he succumbed to an infection which otherwise should have been safe. Just a kind of a case report, but illustrating a general point. So we know iron deficiency, which is probably the more familiar version of a more common version of an iron-related disorder, and anemia are incredibly common. Approximately a quarter of the world are deemed to be anemic, depending on which cutoffs you believe. And about half of that is thought to be due at least primarily due to iron deficiency. And the 2017 global burden of disease estimated that about 1.2 billion people suffer from iron deficiency anemia, and about the same number again perhaps have iron deficiency without anemia. Anemia is a relatively late stage manifestation of iron deficiency. And in most of sub-Saharan Africa and in many South Asian countries, it's the leading cause of years lived with disability. And we know that this influences growth in utero and also during infancy, but also affects adolescents and the elderly too. So iron deficiency anemia, very well known, treated with oral or IV formulations as well, and the iron supplements are cheap. The planet's full of iron, so what's the big problem? And as you'll know, if you're in the iron field, the problem is infection, and this was illustrated most dramatically by the Pemba trial that we were discussing briefly this morning in Santa's lab meeting. Pemba off the coast of Tanzania, a site of an enormous clinical trial, 24,000 young children involved in this, where iron folic acid or iron folic acid and zinc or placebo were given to 24,000 infants. And the trial had to be stopped after 11 months by the Data Safety Monitoring Board because of increased numbers of children who were getting malaria or other infection-related events, either deaths or hospitalizations. So a trial that was meant to show a long-term benefit of iron in an area with a lot of anemia was stopped because of an unexpected side effect on infection. So a kind of a classic own goal. The analyses of cause-specific admissions to hospital and deaths confirm malaria and other infections, infectious diseases significantly increased in children given iron and folic acid. So lots of other work has gone into this, lots of other clinical trials, genetic evidence and so on, and experimental work indicating a link between iron and infection. So the questions that really arise from putting those slides together is, one, why is iron deficiency so common? And I've got a slightly different take on that. Why is iron so important during infections? And then because we're talking about infections, we have to talk about immunity. How does iron influence the immune response? And these are the three things that I'll be talking about. Okay, so the take that we have ended up getting on really the first two questions, which will be the kind of the intro to the immunology, really goes back to the origin of life. Thought to be about 4.2 billion years ago, although nobody's sure because nobody was around. But we also know that there was essentially oxygenless atmosphere at that time. There were little whiffs of oxygen, but almost no oxygen was around. And in the absence of oxygen, iron is very soluble in water, and that's important. So it's very abundant in the planet, and as Fe2+ then was potentially bioavailable to emerging life. The other key biochemical property of iron is that under the conditions on Earth now and then, it's very easy for it to donate and receive one electron, so it can shuttle between oxidation states, and that's critical for thinking about its role as a catalyst or involved in catalyzing chemical reactions. Not only that, it's quite promiscuous. It can hybridize its orbitals, and it can bind lots of different elements in different orientations. So it's not particularly restricted in what it interacts with. And these three characteristics are unique to iron. There is no other element that can shift valency very easily under the conditions of Earth, is also bioavailable and highly abundant, and can bond with many other elements. It is only iron that is like this. Not to say that other metals aren't important in biology, of course they are, but iron is a special case in this respect. At least I hope it is, otherwise my career is based on really not very much, is it? So here's two examples, an iron-sulfur cluster binding to cysteine, and of course iron in the middle of heme. And you can see that they're really pretty different things, and they're both very important in biology. But those are just two examples of iron in particular cofactors, but iron also independently can bind into many different proteins in different orientations too. And a whole lot of work has gone into thinking about the role of iron in early life, and a consensus is emerging that iron helped biochemical processes that were to evolve from anyway ongoing geochemical reactions. Biochemistry had to happen, had to come from something. And there was geochemistry going on that really resembled very closely basic biochemical processes now. And in fact, you can get almost all the way around the Krebs cycle just using iron as a catalyst without any enzymes at all under the right conditions, which is a remarkable finding. So electron transport chain in the Krebs cycle, ancient and iron dependent. Very ancient processes in simple forms of life, like nitrogenases and hydrogenases, are always iron dependent enzymes and have been for, as far as we know, 4 billion years. And making DNA, a lot of the enzymes that are involved in DNA metabolism, but particularly the rate-limiting step of making nucleotides, ribonucleotide reductase, is an iron-dependent enzyme. And in fact, now in humans, about 2% of our genome is a code for iron-interacting proteins. And iron isn't performing a structural role here. It's almost always catalytic. It's almost always a critical enzyme in a critical biochemical pathway. So in mitochondrial metabolism, DNA synthesis and repair and initiating replication, enzymes involved in epigenetic modification, making DNA, and many, many others, oxygen transport, hypoxia sensing, killing bacteria, making heme and detoxification enzymes. These are all conserved and iron-dependent kind of right across life. And in fact, Otto Warburg, who you might know from the Warburg effect, but in fact he got his Nobel Prize for showing that iron is a critical component of respiration, said that life without iron is impossible. There are 2 exceptions to this, in fact, but only 2 exceptions that we know about. So still, this is pretty cool. So iron became indispensable for life at a very, very early stage. All these basic cellular processes need iron and still do. And that was fine at the time because there's a lot of iron around and iron is soluble in water. So it was very easy for life, simple life, to assimilate iron from the environment at that time. But then photosynthesis came along about 2.5 billion years ago. And probably originally in marine cyanobacteria. And the point about photosynthesis is the waste product, as it were, is oxygen. And oxygen has a very close and very important interaction with iron, which of course we use in our red blood cells to carry oxygen around. But at the time, this was pretty disastrous for the life that was around. And this made iron insoluble. So all of that nice iron, easily available to life, became relatively insoluble very quickly. And you can see this in the geological record, banded iron formations. They're all over the planet, and this is now iron still abundant, but no longer bioavailable because it's locked up in rocks. So this key nutrient that I've banged on about being fundamentally required for metabolism and growth was relatively abruptly very scarce. This is a huge selection pressure and actually led to an enormous extinction event too. And in fact, you can think about planetary hypothermia and map it over time. We've done this by collaborating with geologists and doing some maths I didn't understand at all. But you can essentially see that, I think I'm on there now, yeah, that originally very high levels of soluble ferrous iron plummets all the way down to where we are now, being almost essentially absent from water as the amount of oxygen goes up in the great oxygenation event there and something called the Neoproterozoic oxygenation event later. And there's also a big drop in ferric iron as well, which is relatively insoluble anyway. So very little iron is available from the environment. And this is key because it means now, where do we get our iron from? We don't get our iron from the environment. We get our iron from things that already have iron. In other words, when we eat. And that's kind of a fundamental change in life. Life doesn't get iron generally from its environment anymore. It often gets it from other forms of life. So although, the reasons of diet, there are reasons of inflammation, that it's important for why iron deficiency occurs, in a way, the basic reason is the oxygenation of the atmosphere is why iron deficiency is the most common micronutrient deficiency in humans. It's because iron is insoluble, it's difficult to get from the environment. But the importance of iron is shown that life is still addicted to it. You can't evolve out of needing iron. And it's sort of trivial to say, but iron isn't an amino acid, it's not a vitamin, it's not glucose, you can't synthesize it. So it's really hard to gather from the environment. And in fact, to get it, you have to steal it. And this is what infection is. Infection is essentially the thieving of nutritional resources from the host by the pathogen. And in particular, iron acquisition from the host is this critical virulence factor, reflected in that case report of Professor Kasadaban and the results of the PEMBA trial. If there's enough iron around, then that can change the outcome of infections. So that means it's not easy and not always safe to increase iron, even if you're trying to combat iron deficiency and anemia. And also, importantly, hosts have evolved to defend their iron strongly, and that's an ongoing arms race of trying to acquire iron and trying to defend it. So in humans, as you'll know, hepcidin is a hormone that regulates iron homeostasis, and interestingly, it's genetically related to antimicrobial molecules, already showing this deep underlying link between iron regulatory hormone and antimicrobial defense. And it's primarily secreted by hepatocytes, but it's increasingly clear it's made by immune cells, and that's important too, so dendritic cells and keratinocytes, but also the heart seems to make hepcidin as well. And just in brief, because you guys all know this very well, we know how hepcidin controls both the absorption and the flux of iron. So we absorb about 1 milligram of iron per day from the diet, loads onto transferrin, transferrin delivers it to where it's needed. And in us, in steady state, most iron goes to red blood cell formation in the bone marrow. But for example, in infants, it's thought most iron will probably go to the brain. And under other conditions of an ongoing massive immune response, that needs a massive amount of iron, and I'll come on to explain that later. But most of the time, the iron is in our red blood cells, which have a finite lifespan, after which they're taken up by subsets of macrophages, which chomp up the red blood cells and release the iron back into plasma. And more iron is recycled per day than is absorbed. And we know ferroportin is the only known mammalian iron export protein, and we know that hepcidin is the hormone that controls and blocks ferroportin. So the amount of hepcidin then determines how much iron there is absorbed into the system, but also where it is in the system too. And we now have a crystal structure of ferroportin, a nice transmembrane protein, and hepcidin very satisfyingly just sticks in the hole and stops ferroportin from exporting it. So it works how we all thought it would. It just blocks the gate. And in the kind of iron field, the endocrinologists, as it were, like to say hepcidin is to iron as insulin is to glucose. And that's both correct and right, but also an important simplification because it turns out hepcidin has a split personality, and it's not just a hormone, it's also an acute phase response protein. And this is really important from thinking about infection and thinking about its origins and thinking about what it does to immune responses. So infection and inflammation stimulate high hepcidin. So in this state, we have a steady state occurrence of iron being recycled from red blood cells and iron being absorbed from the diet, coming through ferroportin and being loaded onto transferrin. If into this situation an infection is added, then infection is sensed by the immune system, and part of the innate immune response is to make this avalanche of cytokines, one of which is interleukin-6, which switches on hepcidin. Hepcidin then blocks ferroportin, and eventually this leads to a dearth of iron in plasma, this hypothermic state, which is acute but also profound, and you can get down to transferrin saturations of 2%, very, very, very little iron detectable in the blood. We've observed this in many infections in humans, and obviously it's observed in preclinical models as well. This is the hypothermic state. It's very common. So it happens in nutritional iron deficiency, but also happens in chronic inflammation and in acute infection. And this is vital for defending against some infections, particularly those of extracellular bacteria. And this is the mechanism that was essentially lacking in Professor Casadaban. He had hemochromatosis, had hepcidin that couldn't be upregulated enough to defend his iron from the invading pathogen. But we know that in preclinical models, this hepcidin-induced hypothermia is absolutely critical, life or death, against infection with particular extracellular bacteria. And the mechanism is very simple. You just take the iron away and the bugs don't divide. And it's as simple as that. But it's not as simple as that for other types of infection, as I'll come on to. And it's not as simple as that in thinking about what could it do to the host. So this low serum iron is very highly evolutionarily conserved response to infection, happens across vertebrates, and is due to high hepcidin, which lowers plasma iron. And this is a part of what has been termed over many decades nutritional immunity. It's probably one of the most important aspects of nutritional immunity. But what happens to the host? I just told you that all of our cells need iron. It's not just the bugs that are invading us, it's also all the other cells that we have. So what is the effect of not having iron available to all those other cell types if there is no iron in plasma, which is the source of their iron mostly? And in particular, you can get very, very, very severe hypothermia in profound inflammatory conditions. So this is very severe COVID, for example. So in this graph, the usual range for plasma iron in adults is from 10 up to about 30. And in here, in the relatively non-severe hospitalized COVID patients, they're already very hypothermic, but the most severe and ventilated patients had essentially undetectable plasma iron for weeks. This is not going to be good for many of the cells to have essentially no form of iron. So what are the effects on the host? And there are probably many, there are probably multiple, but I'm just going to be really talking about lymphocytes. So lymphocytes really need iron too, and this has been known for a long time, but has become very clear recently. So when a lymphocyte is switched on, probably the protein which is most upregulated is actually the transferrin receptor. It's enormously upregulated within 24 hours of activating a CD8 T cell. It goes from almost undetectable to well over 1 million copies of protein per cell. And at the same time, interestingly, ferroportin, although there's not much ferroportin on T cells, it's switched off by orders of magnitude. So activated T cells become iron scavenging machines. They're desperate to get as much iron as they possibly can. And this is because they actually begin proliferative programs to divide every 6 to 8 hours, have to replicate everything. They've got to do enormous amounts of macromolecular synthesis, and they've got to reprogram their metabolism. They need an enormous amount of iron to do this. So as you might expect, if they don't have enough iron, this can be a problem. And I think that this really, people started thinking about this, and with this particular paper, which was really remarkable and came out to a lot of people's surprise, when a very rare series of mutations in transferrin receptor were observed in only 8 patients from the Middle East, from consanguineous families. And I think if this particular mutation causes transferrin receptors to be a bit less good at taking in iron. It's not a knockout. A knockout is lethal. It's about a 50% effect on transferrin receptor being able to get iron from the extracellular environment and put it in the cell. And I think everybody in the iron field would have predicted that the phenotype would be anemia, because red cells need iron, right? These children are basically not anemic. They've got hemoglobins about 10, 11, 12, maybe a little bit more. So they're kind of borderline anemic, but they are profoundly immunodeficient. They can't mount T cell and B cell responses to either vaccines or pathogens, and they had recurring infections that had to be cured by bone marrow transplantation, except in those children that they didn't get to quickly enough who died. So really profound. Immediately raising the issue that iron acquisition by lymphocytes is actually astonishingly and unappreciatedly important. So if you have a wild-type transferrin receptor massively upregulated on a T cell, that takes in iron, you have immunocompetence. If you have a mutation in transferrin receptors, so it's not as good, this diagram is a bit too dramatic. There should probably just be about half as many red dots in this cell, not no red dots, then you're profoundly immunodeficient. So imagine then a situation where transferrin receptor is fine, is wild type, but in fact, there's no iron available in plasma, that hypothermic state that I was discussing with high hepcidin or severe nutritional iron deficiency. So could hypothermia then inhibit an immune response? And that was the hypothesis that we were kind of getting going when Santa was just about leaving the lab, and that we've run with since. And this is the major paper that's come out. There's the foundational paper for our lab doing many of the things in this now, showing basically that it is the case. So I'll walk you through the critical experiments that are published, and then a bit more that's resulted from that that we're following up on. So we can obviously control plasma iron in animals very profoundly and very quickly using hepcidin, or we use a mimetic called mini-hepcidin. So we just inject this mouse with this particular molecule, and very rapidly, within a day, within hours, and lasting about a day, plasma iron drops right down. But you'll notice that this level here, although it's really low, it's actually not as low as those COVID patients, and it's not as low as somebody with humans with controlled infection with typhoid, or people with severe nutritional iron deficiency in the Gambia, for example. So it's really low, but it's not unphysiologically low. So the setup of the experiment really is to just jab a mouse with a vaccine or some other immunological stimulant, which doesn't by itself alter significantly the normal range in mice. And then at the time point where they were developing a T cell or B cell response to the vaccine, or the stimulus, to then put them on a daily injection of mini-hepcidin, either on a background of a low-iron diet, but it turns out that that isn't needed. Mini-hepcidin alone is sufficient to do this. And the result was very clear. So you kind of completely crush the T cell response. So this is tetramestaining of antigen-specific CD8 T cells to this particular adenovirus vaccine. And each dot here is a responding cell, and there are basically none in the mice that have had their iron manipulated. And then in here, this is each individual mouse. And so you can see that the mice look as if they basically haven't been immunized at all, but they have. They've just had their plasma iron suppressed. And this is a different vaccine with protein in adjuvant. And we've observed similar inhibition of the CD4 T cell response and the B cell response and the production of antibodies. So the whole adaptive immune response is really severely crushed by altering the immunological, the availability of iron through hepcidin manipulation. And the scheme overall is that, when a dendritic cell or some other, usually a dendritic cell, stimulates a T cell, or if a B cell is activated, then it massively upregulates transferrin receptor, obtains the iron it needs from plasma, and then proliferates like crazy to make a really big clonal immune response against the antigen or the vaccine or the infection. But if the mouse is injected with hepcidin, then transferrin receptor is still upregulated, but there's little iron available. And as a result, the proliferation falls over. The cells that are produced are a bit rubbish. They just kind of, we imagine them as kind of bored teenagers that just sit around and they don't really do much on the sofa. And they don't make many cytokines. They're functionally just not there. And we've observed this in blood, spleen, and lymph nodes, and we've observed it across a whole range of different types of vaccines. And I'll come on to a particular vaccine that we've used, which has been very interesting for thinking about planning trials related to this. But one of the things that we wanted to think about was, okay, these are our vaccines, which are often based on viral vectors, but what about a real virus, and what's the response there? So we then tested, what about immunity to influenza virus? So we gave mice intranasal influenza virus and then gave them the daily hepcidin injections and then measured the immune response, so very similar. And again, we could see a severe inhibition of the T cell immune response, CD8 T cell response to the vaccine. And again, this now is just hepcidin alone, not the low iron diet. A decrease in the CD8 T cell response in the spleen and in the lung. And even more severe suppression of the germinal center B cell response, which are really proliferative iron-requiring cells. The germinal B cell response is often just not really detectable at all by flow. And there were no detectable neutralizing antibodies in the mice given flu and mini-hepcidin. Neutralizing antibodies are very important for fighting viral infections in general and influenza specifically. And this meant that the virus couldn't be controlled, so we had greater persistence of viral RNA in the lung and much more damage as a result. So increased histological lung inflammation, particularly with some neutrophil infiltration, but there are other cell types as well. So the disease was definitely worse as a result. And when you looked in the lungs, this is what a lung should look like. This is what a lung looks like with flu. And then with flu and mini-hepcidin, it's an absolute mess and just didn't look like a lung at all. And we had stains for various different fibrosis markers and so on like that, which lit up a lot. So persistent hypothermia is worsening in influenza virus infection. But you recall from earlier on in the talk, I was saying that the same thing, hepcidin and low iron, protect against extracellular bacterial infections. So this is the opposite of flu. So what's going on here? How can we try and resolve this? So our current theory is that inflammation downstream of infection events or other events switches on hepcidin via IL-6, and hepcidin then regulates the amount of iron in plasma. If the cause of the inflammation is an extracellular siderophilic pathogen or potentially malaria, although I'll come back to that, then this is useful because it takes iron away from the pathogens as a benefit to the host. But if the cause of inflammation is a pathogenic virus, and viral replication does not have the same sort of iron requirement as a bug trying to replicate itself, it has some, but really nowhere near as much, then this could be a risk to the host because you're taking iron away from the immune response. And the relatively defective immune response means that the virus isn't controlled as well. You get extra viral replication, as we've seen in flu, and we've seen that in a different viral infection where the controlling element is an NK cell. And this leads to more profound inflammation and an even bigger hypothermic state, and potentially even further defects and risks to the host physiology. I've mentioned malaria there, and I just put in an extra slide at the last moment, just based on what Danielle was saying in Santa's lab meeting. So there's a really complex relationship between iron and malaria. Just to say that we know that iron deficiency is caused by malaria, but iron deficiency also protects against malaria in ways that we don't fully understand. But we know that it's at least partially mediated through erythropoiesis, but also potentially through effects of iron on less iron in the liver and in red blood cells to supply the parasite growth. However, two really important other aspects of malaria are tolerance to the parasite. So does the parasite itself cause disease or not? That is actually independent of, it seems to be independent of the amount of parasitemia. It's something that's intrinsic to the host. And also the immune response to the parasite, which is partially related to it. And we still don't really understand how iron relates to these important things of disease tolerance and immune response. We have some preclinical data on that, but I think this is an important extra thing to behold and think about, not only with malaria, but also with things like COVID-19, where there's a lot of immunopathology. How is the iron actually affecting the immune system going out of control? That's another kind of frontier. Okay, how are we then beginning to move this concept to think about translation to humans? And one of the experiments that we did that led to this was we did an experiment where we thought we'd test our effect on what was at the time the developing and just being released Oxford AstraZeneca vaccine against COVID. So we used the same platform, this ChAdOx1 virus, but not encoding a COVID antigen. We thought that might be a bit too much on the nose. Instead, we did it with ovalbumin, and again tested what would happen with mini Hepsin, and it absolutely smashed it. It really stopped the CD8 response. We looked in many different ways, and we also looked at T follicular helper cell, and again germinal center response. Adaptive immune response to this very important vaccine platform suppressed by hypothermia. But what we then realized was that there's something nice about it, and one of the reasons why this vaccine platform is so good is that it continues over time to provide persistent antigen. It keeps on making antigen over a long period of time. Whereas we know that these mini Hepsin injections are relatively transient in how they affect iron. They take iron right down, but eventually with a half-life of about a day or so, plasma iron just recovers and goes right back up. So we wanted to ask, okay, how does that affect the response to the vaccine? And we saw quite an interesting effect where if we did look at day 7, we could see again, if we took a blood sample from the mice, we could see really profoundly suppressed T cell response to the vaccine. But then if we just in the same mice looked at 7 days later, it completely recovered. So the immune response completely back to where it ought to be at that point, which raises lots of interesting immunological questions. So, for example, are these the same T cells that are primed, but then wait until there's enough iron and then proliferate? Or is it an entirely new cohort of T cells that are getting primed once the iron is available? Either way, it's indicating the immune system is able to wait for the right nutritional environment to get activated, which is kind of quite a cool concept. But the other thing that it immediately made us think about was that in an iron deficient situation, can nutritional iron or iron supplementation affect and improve the response to a vaccine? So we tested that immediately in mice, very simply by transferring antigen-specific cells into a mouse, priming the mouse with that antigen, and then giving the mice either mini-hepcidin or mini-hepcidin with iron to counteract that suppression. And saw a very nice response where we could block the T-cell response with mini-hepcidin, but recover it if you gave iron back into the system. So that was the first way of doing it. The second way of doing it was in piglets. So you might know this, but every bit of bacon, if you're a meat eater, every bit of bacon you've ever had has come from a piglet which has been injected with iron at birth or been given a lot of iron supplements. Because over many centuries, pigs have been bred to have very big litters, many animals, and for them to grow very quickly to provide pork. But as a result of that, they grow so fast, there's not enough iron in sow's milk to support them. So in order to kind of get through that growth spurt, they need extra iron. And if they don't, they get anemic really quickly. So in mice not given that iron, we deliberately withheld that normal treatment, and then these piglets have very low serum iron and do become anemic, whereas the ones that were given iron had higher iron and higher hemoglobin, but they also had a better immune response to the normal veterinary vaccines which are given to piglets at that point in time, showing that this nutritional iron to counteract iron deficiency anemia also improves the immune response in this preclinical model. So that obviously means maybe against a background of iron deficiency, which as you know is so common, can we improve immune responses to vaccines? And we know that the vaccines often don't perform very well in populations where iron deficiency is common. So we've got 3 ongoing human studies, one in Kenya, in infants, one in pregnant women on the Thai-Myanmar border, and another one in Kenya, which has actually done IV iron in response to COVID, flu, and yellow fever in anemic Kenyan women. And these are going to provide hopefully a whole load of data which will allow us to kind of combine hematological responses with immunological responses. The idea that iron is not just a red cell thing, it really is a white cell thing too. And so we're doing throwing the omics at these trials. Okay, so some unknowns that result from all of this. So why is iron so particularly important to lymphocyte physiology and fate? What's the kind of the metabolic basis of this, and how does it affect lymphocyte decision-making and differentiation and so on? Something more about quantification. You know, we kind of define iron deficiency by proxies of iron. We don't actually measure the amount of iron in cells, but that's the thing that actually matters. So we need to get better, I think, at quantifying what iron deficiency means in terms of raw atoms of iron in cells, and we're getting technology to be able for us to do that, working with the geologists. What about other leukocytes? So they're affected by iron deficiency. It's not just all about lymphocytes. And then finally, you know, can we weaponize this usefully? You know, we don't want to be going around inhibiting vaccines. Can we therapeutically manipulate immunity in a useful way using iron? And in the remaining hour and a half of my talk, I'll just cover these, and don't worry. We have been making progress on some of these. I'll just briefly tell you where we've got to on a couple of them. So other leukocytes affected by iron deficiency. We did some data mining, and we were able to show that in terms of per cell, that neutrophils ought to contain much more iron than any other type of white blood cell. And this was based on some combining different data sets that were already out there. And this was because a lot of the neutrophil granules that are used for killing bugs and so on are iron-dependent enzymes like myeloperoxidase, for example. So that's basically why that might be. But of course, neutrophils, you also make an absolute ton of neutrophils per day. So if you combine the amount per cell with the amount of cells that are made per day, and then say, okay, milligrams of iron per day, what's needed? It turns out that neutrophils are only about 10-fold less than red blood cells, but neutrophils have a much shorter half-life than red blood cells. And so this made us predict that perhaps if you were in a low iron state, then the leukocyte production that would be most affected most quickly would actually be making neutrophils. And this turned out to be true when we tested it experimentally. So again, we just jabbed mice with Hepsin and then just went across the hematopoietic tree and looked at different cell types, and just showing that bone marrow neutrophil production and eosinophils, which are also quite iron heavy, were really suppressed by hepcidin, but other cell types not shown here just weren't, including monocytes. And in the blood, this was the same story. We could see a big suppression of neutrophil production and eosinophil presence in the blood, but monocytes, dendritic cells, T cells, B cells really just didn't care about that. They could carry on being made. And this in fact is consistent with that transferrin receptor mutant paper. Those children with mutations in transferrin receptor, they had normal levels of circulating lymphocytes. So the hematopoietic production of those lymphocytes was okay. It was what happened to them after they were activated. They then couldn't proliferate. And those children were neutropenic as well, again, consistent with this, that it's neutrophil production that is particularly iron hungry. And you could show this we could also rescue this by iron supplementation. So you can deplete neutrophils with an antibody called anti-GL1. And then while the bone marrow is trying to reconstitute, you can prevent that by injecting the mice with mini hepcidin. But then you can counteract that by giving them ferric ammonium citrate, and all the neutrophils come back. So that was kind of a really nice experiment. And the transferrin receptor expression is a marker of iron deficiency in the progenitors. They were massively iron deficient in the hepcidin-treated mice, and then that returned to normal in the mice that had been given iron. In neutrophils that did come out from an iron-deficient host, they were rubbish neutrophils, so they weren't so good at making reactive oxygen species. They weren't as good at actually killing phagocytosing bugs, and they weren't as good at killing bugs. The bugs survived better, so you were making fewer rubbish neutrophils. So this was an important functional correlate. So overall, we could say that this iron deprivation and its effect on the immune response was quite interesting. We showed a clear effect on neutrophils. With collaborators, we saw a big effect on NK cell proliferation and the ability of NK cells to fight a viral infection called Frenz virus. And in the adaptive immune arm, we could show very clear effects on B cell and T cell proliferation and function. So knowing that, then we wanted to know, can immunity then be therapeutically manipulated via iron? And so this is just now a few very brief slides showing a proof of principle on this. So what we go for pretty high bar to try and attain proof of principle. So we've gone for total MHC mismatch of skin transplant. So one of the most severe immune challenges that you can get to transfer skin. And initially, the way that we did this was to get an immunodeficient mouse host on a black 6 background and put a CBA skin graft on. And then into that mouse, you can just inject 10,000 wild-type CD4 T cells, and that's enough to reject the grant. I always say that. It's like some sort of Freudian slip. We haven't yet had a grant rejected on this, but that's because we haven't written one. But I'm just waiting for it. The graft is rejected by the wild-type black 6 T cells. So into this, we put instead also as a second arm, same number of T cells, but from the mouse carrying that mutation in the transferrin receptor that was associated with immunodeficiency. Again, these T cells are completely fine, apart from the fact they aren't as good at getting iron in by a factor of about 50%. And what we found was a very, very clear result. Is that essentially that graft completely survived. There was the same number of T cells, actually, that we could detect in the peripheral blood of both of these mice, but they were just functionally unable to reject the graft. So that was a very kind of remarkable finding that these allogeneic total MHC mismatched T cells, because they had a 50% defect in iron acquisition, could no longer reject the graft. And if you actually looked at the graft itself, then the graft score is showing how kind of grotty the graft is looking. In the wild type, they get grottier and eventually rejected. And there's this interesting sniff of initial reaction here in the mice injected with the mutant T cells, but then it settles down and they're completely fine. So then we went for broke. What about in fully immunocompetent mice? So this is now really just getting a bit of skin from a wild-type CBA and putting it onto a black 6, but doing it in mice which either had a normal iron diet or had a low iron diet and were getting daily mini hepcidin injections. And we monitored the immune response at the same time as looking at graft survival. So we could completely block the immune response to the graft in the mice that were given the graft and on the low iron diet. So this is CD4 and CD8. So this is not an antigen-specific response. This is an allo response to a total MHC mismatch. It's about as high a bar as you can get, and it's the skin, which is very, very antigenic. So huge effect here. And in fact, one of the things that was interesting was that we saw a few more regulatory T cells as well in the mice which had the graft in the low iron. The effector T cell to regulatory T cell ratio was reversed. And we looked at the graft itself. Then while we were able to maintain this low iron diet and the mini hepcidin treatment, then there was very little rejection of the graft. But interestingly, when we had to stop doing that because of animal license rules, then the graft was not tolerized. It was still immunologically able to cause an immune response and then for it to be rejected. So it's delaying but not preventing graft rejection. Nevertheless, this is kind of proof of principle that very, very powerful immune responses can be prevented by mucking about with iron availability. So it kind of opens the door to a whole load of other approaches. Okay, so to summarize, what we've shown is hepcidin is an iron regulatory hormone and acute phase response that we know about, and that controls supply of iron to plasma, and so because of that, to immune cells. And T cell, B cell, and NK proliferation and function require iron via transferrin receptor. So it turns out they are very highly sensitive to low plasma iron and to the iron regulatory hormone hepcidin. So hepcidin turns out to be an immune regulator as well as an iron regulator. And neutrophil production and function is also very sensitive to plasma iron availability, particularly amongst leukocytes. And overall, those findings, I think, inform a couple of interesting things. So a key thing in immunology is to try and understand how the immune system in humans varies under physiologically relevant conditions like chronic inflammation, nutritional variability, including iron deficiency, and so on. And this really adds to that. It suggests potentially new ways to control immunity therapeutically by controlling iron. And in terms of global health and vaccine efficacy, it adds something else to the idea that anemia and iron deficiency and predisposition to infections, and particularly vaccine-preventable infections, might have much more of an overlap than previously was recognized, and there might be a functional and addressable overlap there. So that's it. I'd just like to thank my lab, really, and Joe Frost was the guy who really led that work conceptually, but with lots of help from others, and we've had some great collaborators in Oxford and also beyond in Gambia and Porto and so on, and our funders, including the Gates Foundation and the MRC particularly. Thank you very much.
Thanks. Oh, is this on? Anyway, thanks, Hal. That was amazing. What a tour de force. And I think everyone can now see why we work on iron, because it's just so fascinating. And you can really, it just covers all of biology, but actually all of geology. Well, obviously geology, but evolutionary, the history of the planet. I think that's something we need to add to our lab, actually. We haven't got anyone doing that at the moment. Are there any questions? Ivo, well, why don't you come up here? And then
fantastic talk, really, really interesting and stimulating. A question in terms of the cellular responses. Do you have any indication whether low iron affects memory formation of immunological memory?
Yeah, we have a PhD student looking exactly at that, and the indications are that it doesn't seem to. So we've done that particular experiment with 2 or 3 different vaccine conditions, and it seems to depend a lot on the individual vaccine. we've definitely got evidence that it can, and that's published in the paper. But then we've looked at other vaccines preclinically, and that's less certain. And we're now digging down into the reasons behind that. I mean, I think it has to all come down to understanding better what low iron does to T cells in terms of their reprogramming and their expansion and then their contraction.
But is that dependent on the low iron state being transient or chronic?
Yeah. We've done even transient iron, low iron, did affect memory formation in one vaccine type, but then not in another. And we are at the point of not understanding that, to be clear.
Yeah, Hal, thanks so much. Really good talk. So a couple of questions. With the model where you give these mini hepsidins, and then you realize that cells are unable to proliferate and respond. If you take those cells specifically, put in a mouse, that's perfectly fine. I'm assuming those cells would start responding. You get it?
We haven't done that experiment, although we did consider doing it. What we have done is we've shown it with multiple different ways that you can make a cell iron deficient and it can fail to proliferate in vitro, and it just doesn't proliferate. Then as soon as you add iron back, it takes off again. So but the question is really, are you doing something under iron deficiency which sticks that cell in a particular state? And we suspect that in a real animal that probably does happen because you're affecting epigenetic programming, because all these demethylases and in fact the alpha ketoglutarate are iron dependent. Production of alpha ketoglutarate is iron dependent, and the enzymes that use it to demethylate either histones or DNA are iron dependent enzymes. So we suspect there could be long-term effects on cells, but that is something that is currently ongoing that we're really looking at.
And with all these effects, as you've shown, and they're transient, seem to be transient effects. So when you have a pregnancy where a mother has, usually not in revamp because we give them iron, but usually there's not a lot of iron bioavailability. Do you believe that is then changing the immune system of a fetus that is not having iron, or do you predict that there are ways of counterventing that?
It's a really great question. I wonder particularly actually about placental immunology, because in my understanding at the moment, the consensus is that the placenta takes the iron first, and then that's the most iron hungry organ, and then the growing fetus, and then the mum is kind of like third in line for the iron. So I guess what we're talking about is whether the effects of low iron are going to primarily affect fetal immunology or placental immunology or maternal immunology. And that's obviously pretty tricky to figure out. I mean, we're doing a trial in anemic pregnant women on the Thai-Myanmar border, where we're giving oral iron, but at the same time, those women are getting maternal immunization, either tetanus and COVID. So we're going to be measuring the immune response to those vaccines and tracking the antibody transfer to the baby and following that postnatally. So we'll begin to get some idea, but it's an open question, really.
Thank you.
Thanks, Hal. There's a couple of questions online, but it's 1 p.m. and I think there's another whole seminar waiting to come in and evict us. So I'm sorry, that was a question from Ian Wicks. I'll get him to email it to you a bit later.
Sure, absolutely.
So look, I guess it just leaves us to thank you for a fantastic talk, and thank you for coming all the way down, and appreciate it.
Thanks very much indeed.